Protoplast culture. Protoplasts are cells without a cell wall, but bounded by a cell membrane or a plasma membrane. Using protoplasts, it is possible to regenerate a whole plant from a single cells and also develop a somatic hybrids. Steps involved in the protoplast culture are isolation of the protoplast. Small bits of the plant tissues like leaf tissues are used for the isolation of the protoplasts. Leaf tissue is immersed in 0.5% macrozyme and 2% Wanazuka cellulose enzymes dissolved in 13% sorbitol or mannitol at pH 5.4. Then incubate overnight at 25 degrees centigrade. After a gentle heating of cells, protoplasts are obtained. These are then transferred to 20% sucrose solution to retain their viability. They are then centrifuged to get a pure protoplast as different from the debris of the cell walls. Fusion of protoplasts. It is done through the use of suitable fusers N, normally PEG, that is polyethylene glycol. The isolated protoplasts are incubated in 25 to 30% of concentration of PEG with calcium ions, and the protoplast shows accumulation, that is formation of clumps and fusion. Culture of protoplasts. MS liquid media is used with some modifications in a droplet, plating or micro-drop array techniques. Protoplast viability is tested with fluorescent diacetate before the culture. The cultures are incubated in continuous light 1002 lux at 25 degrees centigrade. The cell wall formation occurs within 24 to 48 hours and the first division of new cells occur between 2 to 7 days of a culture. And next step is selection of a somatic hybrid cells. The fusion product of a protoplast without nucleus of a different cells is called as hybrid. Following this nuclear fusion takes place, this is called somatic hybridization. And next type is cell suspension culture. The growing of the cells including the culture of a single cells or a small aggregates of cells in vitro in liquid media is known as cell suspension culture. The cell suspension is prepared by transferring a portion of callus to the liquid media and acetate using a rotary shaker instrument. The cells are separated from the callus tissue and used for cell suspension culture. Production of secondary metabolites. Cell suspension culture can be used for the production of secondary metabolites like alkaloids, flavonoids, terpenoids, phenolic compounds, and recombinant proteins. Secondary metabolites are the chemical compounds that are not required by the plant for normal growth and development, but are produced in the plant as byproducts of the cell metabolism. Example, biosynthesis and isolation of the indole alkaloids from Catharanthus roseus plant cell culture. Few examples of industrially important plant secondary metabolites are listed below. Applications of tissue culture: micropropagation, virus-free plants, conservation, secondary metabolite production, year-round supply, hybrid production, embryo rescue, maintain quality, genetic modification.